Mohammed, and for putting together this great conference. This is not actually a typo. I'm going to be talking about what I think of as meiotic and mitotic recombination, but during meiosis. So my lab is interested in the differences between mitotic and meiotic recombination, and in particular, how that translates into whether recombination results in crossovers or not. And we summed up a lot of things that we and many others in the field were thinking a few years ago in a review article published by a former student of mine, Sabrina Anderson. I had this photo because I thought maybe she'd be here because she's now a postdoc in Tom Peters' lab, but I didn't see her name on the schedule. Anyway, the overall take-home message from this is that it is based around the idea that in meiosis, crossovers are generally good. And so in meiosis, you want to do double-strand break repair to generate crossovers because this is how you're going to segregate your chromosomes accurately. But in mitosis, crossovers are bad because crossovers can lead to loss of heterozygosity, or if you have non-allelic recombination, to chromosome rearrangements and so on. And so how do cells regulate things differently? Now, Sabrina's approach was to look at genetic interactions between a set of proteins that we're thinking of as pro-crossover. And of course, there's a lot of proteins required to make crossovers, but we're focusing just on the very end of the pathway, which is the enzymes that cut holiday junctions, and then how those interact with one in particular anti-crossover protein. So the pro-crossover proteins, it was a long time before people discovered the nuclear holiday junction resolvases, but suddenly we have a wealth of them. I'm going to use icons to represent them so you don't have to try and translate all the different species that I mentioned. And these icons actually make sense, at least to me. MUS81 MMS4 was the first holiday junction resolvase to be discovered. And I use this icon because in vitro, it kind of rough cuts the junctions so that they can't be religated. So it's making uneven cuts. And then more recently, another enzyme called GEN, or YEN1 in cerevisiae, was discovered. And GEN, or Gen1, as it is in humans, is a much more precise instrument, as you can see, and it makes very clean cuts on holiday junctions, so they can be religated very nicely. And then a third one, which in Drosophila is MUS312 SLX1. I'm really not going to be talking about that one at all, but you'll see what this icon means because I'll talk about an enzyme that has the same handle here but a different blade. Okay, and then on the anti-crossover, the enzyme I'm going to talk about is the Bloom helicase, originally discovered from Bloom syndrome mutations that affect this gene, cause the disorder Bloom syndrome in humans. And a characteristic of cells from those patients is highly elevated mitotic crossovers. Mostly you see it between sister chromatids, but you also see it between homologous chromosomes. And when you knock out the orthologous gene in flies or yeast or many other organisms, you get the same kind of elevation in mitotic crossovers. So we think of this as an anti-crossover activity normally. Okay, so what are these things doing? Well, typically when I give talks, I show these sorts of models, sometimes kind of step through them and all. In this case, you can kind of ignore all of these structures. I'm going to, in this case, go from, instead of icons for what the DNA is, back to words, and just say there's a double-strand break, there's a couple of key intermediates, the extended D loop and the double holiday junction, and those key intermediates can be converted into non-crossover products through the action of this anti-crossover activity. So Bloom's helicase takes these intermediates, turns them into non-crossovers. And the thickness of the arrows is meant to represent how we think of the sort of preference, the hierarchy of what's happening here. I think probably the most common is this process we call SDSA, which is catalyzed by Bloom's helicase and probably other proteins as well. Sometimes things get through to this double holiday junction that's usually dissolved, also into non-crossovers. And then in the case where you're missing Blooms or something bad happened, you can get down here where you have to cut this structure with those resolvases. And that cutting, at least according to models, can give you crossovers or non-crossovers. Okay, meiotic recombination, well, we kind of start with approximately the same, and we usually draw it approximately the same. Only we make our arrows thicknesses in different places, so that now, of course, there is a decidedly pro-crossover bent to some of these. There's still a lot of non-crossovers. In fact, probably most of the double-strand breaks in meiosis become non-crossovers in most organisms that we've been looking at, we meaning the field. But there is certainly a crossover pathway. Now, studies in yeast have suggested that once you get to this intermediate in the meiotic pathway, it really is somehow resolved specifically down the crossover pathway. Also, these things that seem to be making mitotic crossovers are, at least in Drosophila, perhaps not acting so much in meiosis, but there is in Drosophila, we've known for a long time, a meiosis-specific complex that we believe is a resolvase. Showing that's a little bit harder because it's quite a few proteins here, but hopefully we'll have that soon. So the difference is really is that we're trying to go down this pathway, and we have this meiosis-specific pro-crossover resolvase. Okay, so that's a very simplistic view in a lot of different ways. One of the different ways is that the general paradigm in the meiosis field, based on studies in a number of organisms over a number of years, is that there are really two meiotic crossover pathways. And people refer to the crossovers as class 1 or class 2. So I just call them pathway 1 and pathway 2. And a distinguishing characteristic is that crossover interference, the crossovers from this first pathway exhibit interference and interfere with one another. But there are some crossovers that don't exhibit interference. I would say we don't really know why there's two different pathways, but some speculation is that these are the crossovers that are involved in segregating homologs. And these are from the process that's used to do homolog pairing, at least in many organisms. And then people often define these by genetic characteristics of what things are required to generate these. And for the class 2, they have this pro-crossover enzyme, this halogen resolvase. And the class 1 is often defined by MSH4/5, which is in a sense a pro-crossover protein, but it does that in a roundabout way by being an anti-crossover. And I'll explain that on the next slide. I just wanted to point out that if you go through some fairly diverse organisms, you find that there are most crossovers seem to be from this pathway 1, although there's a significant and easily measurable amount from this second pathway as well. Okay, so what do I mean by this anti-crossover? Well, if we put sort of my meiotic pathway that I had earlier, and we have these anti-crossover activities up here. Well, if you've got these sort of driving everything to non-crossovers, how do you get down to this crossover pathway? You need some way to inhibit these or inactivate them or block them or something. And that seems to be what this, because if you don't, you're not going to get down to crossovers. That seems to be what this anti-crossover thing is. It's thought that this MSH4/5 binds to these intermediates and either blocks or prevents access to the Bloom's helicase or other anti-crossover proteins. And so that prevents these pathways and lets you go down the crossover pathway. Okay, so that seems to work fine in many of the organisms that I showed you. My lab works primarily in Drosophila, and in Drosophila we've got a potential problem in that Drosophila is lacking both MSH4 and MSH5. There are other species that lack MSH4 and 5, like Giardia. We don't really know if Giardia actually has sex. Schizosaccharomyces pombe, the fission yeast, but they seem to use a very different pathway, so they may not need these. But Drosophila and the tsetse fly, which is closely related, Glossina morsitans, they seem to be unique among metazoans in lacking MSH4 and 5. So for a long time we thought that just meant, since MSH4 and 5 defines one pathway, that Drosophila must use some different pathway. But for a couple of years now we've been thinking something different, which is somehow the flies lost to these proteins and they instead recruited something else to do the same job or an equivalent job. Okay, and those something else that we hypothesize is a group of proteins that are called preconditioned proteins. I'm not going to try and define that. It's something that Bruce Baker and Adelaide Carpenter came up with 40 years ago. I'm just going to tell you that that is 3 genes and or proteins, which is REC, recombination defective, MEI217 and MEI218. And we think these might be the same because first they have very similar phenotypes when you look at the mutants, which is a large decrease in interfering meiotic crossovers. They seem to be meiosis specific. We can't find any functions outside of meiosis. And at least for REC, similar to MSH4, we find that among the non-crossovers, the gene conversion tracks are shorter than they are in wild type. Okay, so I will tell you a little bit about the proteins. Turns out all the proteins are in this family called MCM, which is for mini chromosome maintenance proteins. And those proteins kind of have some similarities to MSH4 and 5 as I've described their functions here. And that is they're both things predicted to have ATPase activity. And that ATPase activity in some sense regulates what they're doing in encircling DNA. So MCMs 2 through 7, this is actually from a structure of an archaeal MCM. Archaeal MCMs make a homohexamer. Eukaryotic MCMs make a heterohexamer. of 6 different subunits. And pretty much every eukaryote you look at, no matter how diverse you go, has all 6 of these. And every place where genetics has been done, all 6 of them are essential because they're the helicase that is thought to open up DNA for DNA replication. So something that encircles DNA. MSH4 and 5 has also been shown in vitro to bind these special DNA structures, holiday junctions and pre-holiday junctions, and thought to then regulate the accessibility to the anti-crossover. Okay, so what we're proposing is that these things which are related to MCMs have evolved to do a job similar to this. And if that's true, then we would expect that in Drosophila and Glossina, we may see evidence of them changing their function. So yes, we can see evidence of that in a couple of ways. So whenever we draw trees, no matter what algorithms we use or what options we select and so on, the Drosophila and Glossina MCM8, which is what REC is, always have these long branch lengths, and they always kind of stick out. You know, and we can go even to plants and protists and so on. That's not true of the replicative MCMs. Those are always very nice and tightly clustered. So many of you are probably thinking right away, has there been positive selection? Not knowing much about evolution, that's not a question that occurred to me, but fortunately a postdoc in my lab, Nicole Crown, asked us that question. And my first thought was, well, this is probably something that's not that easy to do. Probably have to do all kinds of algorithms and things. Turns out it's not as hard as I thought. My approach was, I went to my colleague Corbin Jones and said, Corbin, I think I owe you a beer. And that didn't quite work. So far it's been 2 lunches and I think only 1 beer. But Corbin has been looking at these to see if there's evidence of positive selection. And so he's been modeling DNDS rates where a number, like a ratio between DN and DS that's less than 1 suggests purifying selection, and a ratio that's greater than 1 suggests positive selection. So we gave him alignments of the DNA for MCM8 from a large set of insects. He eventually pared it down to just the Diptera and then a couple of closely related outgroups. And our question is, does this branch here, which separates out the Brachycera from the other suborder of Diptera, have evidence for positive selection? Well, so in this model, which allows a different rate of DNDS in every branch, almost every branch gives you a number less than 1. But there are 3 branches that give you a number greater than 1, including the Brachycera branch right here. The others are here and here. We don't really know if those have any significance. We don't have any hypotheses for those right now. But this one fits our hypothesis that this branch would give us evidence for positive selection. Now, Corbin knows what he's doing, so he didn't stop with this model. He's done, I don't know, 20 or something other models. Unfortunately, during this time, Corbin moved his lab. Fortunately, it's to a building that's even closer than where he was on campus, a new building that just opened up connected to my building. And so now that they're settled, hopefully he'll get some time to actually look through all of that. There are other models that aren't as good a fit, and there are other models that are at least as good a fit, but this one actually seems to be the best, not surprisingly. So it suggests that there has been positive selection for REC along this branch. Okay, there's another way that MCM8 has diverged in Drosophila, and that is, so we noted a number of years ago that if you look very broadly again, MCM8 and 9, sort of like MSH4 and 5, are always either both absent or both present. And so we had suggested back then that this suggests that they function together. Drosophila and Glossina are unique in that we easily find MCM8, but we can't find MCM9 in any of those species. 2 papers that came out last Thursday online have demonstrated for the first time that MCM8 and 9 do function together, at least in chickens and mice. Okay, so MCM8 has diverged a lot and it's lost its partner. So maybe it has other partners. Well, as I told you, there are 2 other proteins in this group that have similar phenotypes, which are MEI217 and MEI218. And those are kind of interesting because if you do the usual like blast searches with them, for 217 you find Drosophila glossina, 2 other dipteran insects, and nothing else. And the conserved domain database finds no hits, so it doesn't really tell us anything about what the protein does. For MEI218, if you do blast, you see there's some conservation at the C terminus, and it seems to be across metazoa, although many metazoan groups seem to lack MEI218 orthologs. And then with the CDD, you get this weak partial MCM domain, and it's weak enough that I never really believed it. I thought it was one of those sort of random things, except that REC is MCM8, which is a canonical MCM. And so what Catherine Cole, the student who's been working on this, did is structure-based searches, and she used this algorithm called FIRE. And when you do FIRE, it tells you, yeah, there's a very nice AAA ATPase domain in MEI218. It's mutated, so it looks like it doesn't have ATPase activity anymore. But it's of the type that's found in MCMC termini. What about MEI217? Well, it turns out by folding, that looks like an MCM N terminus. And this is kind of interesting because MEI217 and MEI218 are encoded by overlapping open reading frames on a single transcript. A lot of people, when we say this, so this is the DNA sequence in that region, and the C terminus of MEI217 and the N terminus of MEI218. A lot of people say, well, I bet there's some kind of frame shifting in there. But it turns out if we look at all the Drosophila species for which we can get sequence, which is 19 right now, every one of them has this A conserved here. Even though to have this alanine, it could be any base. And so that seems to be conservation of having to keep these as two separate polypeptides. Okay, so has MEI218 and MEI217 changed from ancestral? Well, in a way they have. If you look at the vertebrate orthologs of these, this is the structure of a typical MCM. You got your N-terminal domain very close to the AAA ATPase domain, and they're on one polypeptide, and typically they're about this size. In Drosophila, these have separated into two polypeptides, and then MEI218 has this very long addition on it that is very poorly conserved. So they have also changed quite a bit. We haven't looked yet to see whether there's been positive selection in here, at least in Brachycera versus other dipteran insects. And I should tell you that there's actually a lot more about MEI218. And in fact, we're not even convinced that these two differences are related to the function that I'm talking about today, although they are still differences. So to learn more about MEI218, let me highlight a poster and a talk. So Catherine will have a poster tonight about this story with more detailed information. And then Victoria Catani is going to give a talk tomorrow about positive selection in MEI218 and Drosophila, and that explaining recombination rate differences between different species. So that will also be very fun. Okay, so we've got these proteins that are related to MCMs, and we have this hypothesis that they form a complex that we think looks something like this. And Catherine has some evidence for this. You can see it on her poster. These two proteins do interact in a yeast-2 hybrid assay. These two proteins, there were problems with the yeast-2 hybrid, but Catherine's expressed them in a baculovirus system in insect cells, and she can do reciprocal co-IPs very nicely. And then I've added MCM5, which is one of the canonical MCMs, and that's based on genetic data right now. There is a single allele of MCM5 that Scott Hawley's lab isolated that's separation of function, so it retains the replication function, but it is meiosis specific and has very similar phenotypes to these others. And Catherine also has some transgene data that says that there's another ATPase subunit in this complex besides REC, and we don't think it's MEI218, we think MCM5 is probably it. So we don't know the exact constitution of what this complex is, but something like this. And so what we propose is that flies, or some ancestor to Brachycera, had a good MSH4/5. It also had an MCM8 and 9 that presumably function together. These are also now known in vertebrates to function in recombination in some poorly defined role, but they presumably had some kind of recombination role. They also had this thing, the vertebrate name is C8 or 45. The ancestor of MEI217 and MEI218. Somehow this became broken. You would think that would lead to chaos because they wouldn't be able to do meiotic recombination. There'd be all kinds of non-disjunction. But possibly this was able to do partial compensation for that job, enough that this animal could get through this problem. But they evolved to, there's no pressure on this, so that got lost. This changed into something else, and its partner was lost. This thing changed into these two polypeptides, which are different shape. All these things came together and made this new complex. Okay, so we can actually test the idea that it's doing the same thing as this anti-crossover role. So the idea is you need to make crossovers in meiosis. Bloom's helicase prevents making crossovers. This complex prevents Bloom's helicase from preventing crossovers. So if you get rid of Bloom's helicase, you don't need this anymore. So Catherine tested that. This is a graph showing recombination frequency across a pretty broad region of the genome. This is about 40% of the genome, 20% of the genome. And in wild type, if we add up I think it's 5 different intervals. That's the frequency of recombination. Rec mutants have a greatly decreased frequency of recombination. Bloom's mutants have a slight decrease. That's something I'm not going to talk about right now. But when you make the double mutant, what happens is the crossovers come back. And this actually rescues the non-disjunction that's seen in rec mutants also. So these mutants have very low levels of non-disjunction. So that demonstrates that, yes, rec really is an anti-crossover thing that's suppressing Bloom function. And we get similar for MI218, except we only have non-disjunction data right now, not actual crossover data. Okay, so this is now the Drosophila pathway I've shown you for the type 1 that I'm calling it, where we've got this thing as our anti-crossover and this thing as our pro-crossover. We always have to compare things to Saccharomyces cerevisiae, which is missing an E on here. In Saccharomyces, we have a different anti-crossover. Recently, some work in Neil Hunter's lab has found that there is a meiosis-specific resolvase, at least a presumed resolvase. There's no in vitro data now, but only genetic data, that is a meiosis-specific pro-crossover protein. So in one sense, these kind of look different from one another, but I think at heart they really are the same pathway. We just swapped in different things. And I'll show you at the end that other species have swapped in other things as well. Okay, so really I think maybe the fly meiotic recombination pathway is sort of underneath really the same as the Saccharomyces cerevisiae pathway. What about this pathway 2? Pathway 2, we kind of draw like this often. Based on Saccharomyces cerevisiae, MUS81 is the major resolvase in this pathway. In its absence, yen1 rather, can compensate for that. Now, recent studies came out a couple months ago in Michael Lichten's lab and Neil Hunter's lab. In Michael's lab demonstrated that non-crossovers in meiosis seem to come from SGS1 helicase, which is the ortholog of Bloom's helicase. Michael's lab has also shown, and Neil's lab as well, that these double holiday junction intermediates can be converted into non-crossovers, again by SGS1 with other proteins acting with them as well. So this, it turns out, looks an awful lot like that first mitotic recombination pathway that I showed you. And so I'm calling this mitotic in quotes. It's not really mitotic, it's meiotic. There's lots of stuff up here, all the stuff I've ignored on this one little arrow here that's meiosis specific. But to me, it helps me think about how recombination may have evolved in meiosis, that one of the pathways is really just a sort of modified mitotic recombination, not too surprisingly. So what about this pathway? Well, we've got the same intermediates up here, so maybe we could draw it really as this is just a branch off the same pathway. So one way to think about it is, yeah, it's 2 ways to get crossovers, but they're just sort of different branches of one pathway. And so we started with a meiotic crossover pathway, or meiotic recombination pathway. We took that and made some modifications, and we added this extra branch that specifically makes crossovers to segregate chromosomes. Okay, so one other thing that came out of these is both Neil and Michael in their papers pointed out that there's a pro-crossover function of SGS1. So I've been talking about this thing as an anti-crossover protein, but it has some poorly understood pro-crossover function as well. And some insights into that, I'd like to think, have come from another experiment that Catherine did. And that is if we go back to the fly ones and we focus on this little part here where we've got our anti-crossover and our pro-crossover thing here. This key intermediate here gets converted into crossovers. Well, what if we get rid of our resolvase here? Well, if we do that, you'd think you get stuck at this intermediate and it would be a problem. It turns out MEI9 mutants are fertile, and so we think they can get rid of this. And we think they can get rid of it just by doing this non-crossover pathway. So then we asked, well, what happens if you get rid of both these? Now we think you'll really be stuck and there'll be chromosomes breaking and so on. Well, Catherine made the double mutant, and it was actually a very surprising result. So this is a graph that's the opposite of what I just showed you. This is non-disjunction. So this is when you have lots of crossovers in wild type, you get very low non-disjunction. When you get few crossovers in MEI9 mutants, you get very high non-disjunction. The Bloom's mutant, again, have a little bit of a phenotype to start with that I'm not going to discuss right now. When Catherine made this double mutant and analyzed them, turns out they have low non-disjunction. So as far as we know, MEI9 is required to make almost all the crossovers, and yet they're able to segregate the chromosomes fine. We haven't measured crossovers yet. That's not as easy as it sounds because these flies aren't all that happy to start with, but we will do that. But presumably this is because they're making crossovers some other way. And there's two ways to think about that. One is hypothesis A, that I'm going to put the Bloom's helicase right here. It's somehow driving things into this pathway. You know, maybe it's recruiting this or the thing that's going to inhibit it or something. So in the absence of Bloom's, everything goes down this mitotic pathway, and we're still getting crossovers from these other resolvases. So of course we want to ask whether these other resolvases give us crossovers in the Bloom's mutant. Unfortunately, all the resolvase genes are essential in the absence of Bloom's, so we have to do some special tricks to do that. Hypothesis 2 is that this double helix junction is really the same as this. It's just that there's all the meiosis-specific stuff that's globbed onto that. Once all that stuff gets removed, you kind of just push it back into what's really the mitotic-like pathway. And now we can do our dissolution. If we get rid of this, then we go through resolution instead. And we have a couple of different predictions that might distinguish those two models from one another. For example, one of them is that if we never go down the meiotic pathway to start with, this mitotic-like pathway is going to give us a different distribution of crossovers, because all that regulation that acts on deciding where you put the crossovers is going to be thrown out the window. We know that once we get down to this step in a MEI9 mutant, the few crossovers we have left have the normal distribution. So if we get down to this step and we put them into this pathway, we should still have the normal distribution of crossovers. Well, like I said, we don't have that double mutant yet, but we do have the Bloom single mutant. And in that Bloom single mutant, we've measured crossover distribution. And if you look in wild type, here's those different intervals I was talking about. And if, you know, crossover rates are different in different intervals. And these are large, like megabase size intervals. So we're not looking at hotspots per se. If we look at the Bloom's mutant, this is what happens. This is the overall frequency across all these intervals. We get essentially a flat distribution. Now there is a caveat to this in that, as I said, you get mitotic crossovers in the Bloom's mutant. So some of these are pre-mitotic crossovers. I think it's a small fraction of them, but we haven't been able to measure that difference yet. Although we got some ideas of how to do that. Okay, so here's what I put together for the fly, the way I'm thinking about the fly meiotic recombination. And that is, at least down on this part of the pathway, you've got your non-crossovers this way. You've got your maybe Bloom's driving things into this, your anti-anti crossover allowing you to make stable intermediates that can then be cut by this meiosis-specific resolvase. And then you've got maybe as a minor or backup pathway this more mitotic-like. Okay, one of the differences I didn't mention, because it's not too relevant, is that in flies, even the mitotic pathway seems to be different in that in some other organisms, this resolvase is the important one, and this one seems kind of mostly like a backup to it. But in flies, this seems to be the important one, and this is relegated to the more backup role. So that's one little flip between things. If we compare this to yeast, well, we've got the traditional anti-anti crossover. We've got these flipped, like I said, and we've got a different meiosis-specific resolvase. But the underlying pathway is pretty much the same. We can compare this to Arabidopsis, and a couple of recent papers have said that FANC-M is the primary or a very important anti-crossover function in Arabidopsis meiosis. They seem to have the traditional MSH4-5. Looks like probably the same meiosis-specific resolvase. I don't think we know about these yet, but I wouldn't be surprised if it's the same. But really, they've just stuck in a different, compared to yeast, a different anti-crossover protein. If we look at C. elegans, C. elegans seems to use only this pathway normally. But if you get rid of this gene called RTEL1, which is also thought to be an anti-crossover protein, then you start needing MUS81. And so RTEL1 could be here, could be here. If you start making extra breaks by doing irradiation, you start requiring MUS81. So it seems like, you know, you might be driving things down this pathway. It still exists, it's just not normally used ever. So the real take-home message in terms of icons is that although these things look like there's a lot of differences on the surface to those of us looking at mechanism, in reality they're all actually pretty similar. And again, probably goes outside of all of this. Okay, so let me just thank the people that have helped with this. So most of this work was Catherine Cole's, and as I said, she'll have a poster tonight. Corbin Jones, who you'll recognize here, is doing the very important work of looking at positive selection. Nicole is one of the other meiosis people in the lab, has made some nice contributions, and then funding from NIGMS. So I'll be happy to take questions now.